very much needed. We are now beginning the first talk of a series of 3 talks on genetics for neurologists. And we have with us Dr. Radhika Dhamija. She's from the Mayo Clinic, Arizona. She's studied from the All India Institute and then has gone and done boards in both neurology and genetics. So we have somebody who can, I've been looking for somebody who can teach neurologists how to use genetics, and I think she's the perfect person to do that. So without much waste of time, Radhika, this screen is all yours.
Okay, thank you, Dr. Kothari, and thank you for inviting me. So I know genetics can be very new to many neurologists. So what I did is I put together a few questions that I'll use a polling to see where everybody is in terms of their knowledge. And I'm starting from very basic, so I can go as detailed or as basic as I feel based on how the poll goes. So there are about 7 or 8 questions that I'll start before I'll go into my talk. So here they go. Hopefully this will work.
For those who have not yet used Slido, either install Slido in your phone and then enter this number N960, or on your phones, open the browser, go to slido.com and enter this number. So if anybody has a problem joining, just raise your hand. We'll give you 2 minutes to join. If there's anybody who has a problem, just let us know. So join at N960. And once you join, you go to the polls. You have to enter your name. Radhika, you may have to reset the poll. Go to the... Yeah. Yes. Okay, so I think most people, nobody's raising their hand, so we can begin.
Okay, so this is the first question. You get 20 seconds for each question. So which of the following methodologies has the highest resolution for detecting a genetic change? Okay, we'll go to the next question. Which of the following methodologies can be performed without requiring a sequence-specific probe? Right? And then Human Genome Project was completed in which year? A 15-year-old girl has a diagnosis of NF1 and has numerous cafe au lait macules, freckling, and neurofibromas. Her mother had only few cafe au laits and no neurofibromas. Which phenomena explains this finding? It's a long question. You only get 20 seconds. What is the most common mode of inheritance of mitochondrial disorders? This is a trick question. A genetic disorder that occurs in males and females equally, yet is rarely seen outside of relatively isolated populations or cultures where consanguinity is common, is most likely. which is most likely explanation of why Huntington disease tends to get progressively worse with each generation? Okay. And this pedigree represents which mode of inheritance? Okay, I think that was the last one. I think you did fairly well, I would have to say that. But I'll open up my presentation now, and then we'll come back to these questions in the end if we have time, or if you have any specific, I mean, the answers are all there. So hopefully you'll all get 100% at the end of my talk. So I have my email on the first slide if you ever need to get in touch with me to ask any questions later if we can't do them in this hour. But the objectives of this talk, we're going to cover really basic DNA structure and some molecular genetics. We'll cover Mendelian inheritance. We'll cover some basic technologies, both in cytogenetics and molecular genetics as it is useful for neurologists. And then we'll spend some time discussing new technologies. So before going into the DNA structure, we'll start with some definitions. So what is genetics? Genetics is essentially the study of specific individual genes and their role in inheritance. And genomics is a study of an organism's entire genetic makeup that is called a genome, and how that interacts with the environmental or non-genetic factors, for example, lifestyle. And genomics is really applied to the study of complex disorders such as cancer or diabetes, heart disease, and hypertension. And we're really not going to focus these disorders for the purpose of our talk. We'll stick to Mendelian disorders. And genomic medicine is a medical discipline that involves using genomic information about an individual as a part of his or her comprehensive healthcare, which could be for diagnostic purposes or even therapeutic decision making. So really, we have come a long way in genetic discoveries. If you think about it, really Mendel's principles of genetics were first described in 1865. And to think about from 1865 to when the first human genome was sequenced was 2003. So the highlights of what happened in this century include the discovery of the DNA structure in 1953. In 1977, there was discovery of Sanger sequencing. In 1985, PCR was discovered. And really after that, the newer technologies, including next generation sequencing, which came about in 2004, followed which the first human genome was sequenced in about 2008. And now we can really sequence any patient's entire genetic information. So this is a cartoon I like to show when we're talking about genomes. So if you think about a book, it is equivalent to the genome. And then the genome is made up of chromosomes, so 23 pairs of chromosomes, and each chromosome, you can consider them as chapters. And then we're talking about gene, you can really equate it to like a sentence in one of the pages. Very simplistic view. So DNA, if you may recall from way back in medical school, it is made up of purines and pyrimidines. There are two purines, adenine and guanine, and there are two pyrimidines, thymine and cytosine. And they're arranged, or they're paired in a double helical model shown here. And there are hydrogen bonds between cytosine and guanine, and two hydrogen bonds between adenine and thymine. Now, the DNA structure that I just described is right over here. So it's a DNA duplex, and it gets coiled really tightly If you remember, these proteins are called histone proteins, on which this DNA is tightly coiled, and this coiling becomes even more tighter and tighter. And finally, you get something that looks like this, which is a metaphase chromosome. So the most condensed phase of mitosis, where you can study the structure of chromosomes, and this is called metaphase. So this is another way of looking at how chromatin packaging occurs in a human chromosome. So you start here from a double helix DNA that I showed in the first slide, and then you condense it over histones to make these nucleosomes and further condense it. And then this is how the chromosomes would look like when they're not in their metaphase. So this is an interphase where they're not condensed. The other things to know is that each chromosome has some structures or names or nomenclature that we will use as I'll describe disorders or technologies. The P arm is the short arm, P stands for petite, and Q arm is the long arm. Telomeres are the ends of the chromosomes. Centromere is where the sister chromatids kind of meet. So I think these few terms are important to know, and you probably already know them. And then again, these are the histone core proteins along which the DNA is coiled. And this histone structure becomes important as we will review some methylation disorders. So how this, again, there are these histone proteins that have these side chains where DNA can get methylated. And so one thing to remember is when DNA gets methylated, that means the gene is silenced, it won't express itself. And again, this becomes important in some imprinting disorders that we'll review in a minute. So reviewing again the basic anatomy of the chromosome, again, there's the short arm or the P arm, the long arm or the Q arm, and then each chromosome has these numbers. For example, you may have heard of say chromosome 15 disorder. So it's actually the 1 stands for the arm, the P stands for the arm. The 1 number stands for region, band, and subband. That's how we know that where in the chromosome an error happened. So deletion happened or duplication happened. So we would read this out as chromosome 15, P arm or the short arm, 1, 2.31. So at region 1, band 2 and subband 3.1, there's an error that happened. This is important to know because that's how you get chromosome reports to look at when you order them. So this is a standard human karyotype. So this was the first test that we all kind of studied in med school. Trisomy 21 or patients with Down syndrome, or trisomy 18 or trisomy 13. This is really how they're diagnosed. So you take the cell and you look at the chromosomes in the mitosis, in the metaphase, which is the most condensed phase of the chromosome, and you look for errors. Say there's an extra chromosome 21 or extra chromosome 13 or 18, and that's how patients were diagnosed with aneuploidy or chromosomal disorders. One other concept that it's important to note as I go in the subsequent slides is the human genome does not only include the nuclear genome that I just spoke about. It also has mitochondrial DNA, and each cell has hundreds and hundreds of mitochondria, and mitochondrial machinery kind of runs on its own. And so it's important to know that when you're looking at a patient's human or genome, then we're talking about both the chromosomes and also their mitochondrial DNA. Now, does DNA get transcribed into RNA? And finally, RNA is processed into mature RNA, and RNA makes proteins. So the human genome is really made up of full letters, A, G, T, and C. And these full letters are arranged in different sequences and condensed in its entirety in every cell. Gene is the basic unit of inheritance, and chromosomes are linear organization of genes. All of us have 23 pairs of chromosomes, and 1 set of each we get from our mom and 1 set we get from our dad. And as I said, genome is all the DNA of these chromosomes within a person. And why it is important to know that we get 1 set from mom and dad becomes important as I will go into exome sequencing. So again, you get one set each from your mom and dad, and that's how a complete set is created in a child. Now, the first test that we all kind of are familiar with is chromosomal analysis. So chromosomal analysis, when you order on a patient, is essentially whole genome coverage. So you are looking at the entire patient's genome and their genetic information, but it is at very low resolution. So only if a change can be detected under the microscope in the metaphase is when you will be able to pick it up. So we say that it is the lowest resolution, even though you can say you're looking at the entire genome. So that's test number one. Now, test number two is something called FISH, or fluorescent in situ hybridization. So essentially what this test is, say you know what you're suspicious about. For example, say you were evaluating a patient and you were worried about DiGeorge syndrome or chromosome 22q deletion syndrome. So there's already a probe that you will create for that site, and you will see how many signals you get in the patient. So as you know, each gene has two copies, one you got from your mom and one you got from your dad. And if one copy is missing, you can say that that patient has a deletion. So this is site specific, though it has very high resolution because you can make probes for a single gene. So we say that FISH has high resolution, but it is site specific. Now, chromosomal microarray, this is obviously a newer test, many years now, but is really routine, at least in pediatric neurology practice, for evaluation of developmental delay or even epilepsy. So essentially, the way I look at this test is hundreds and hundreds of FISH in one testing. So you have all the chromosomes and you have created probes for different sites in all these chromosomes because you know the regions on the chromosomes that are important. And you compare the patient's DNA with a reference DNA and see if there's a signal increase, so duplication, or a signal decrease or deletion. And I have better pictures or bigger pictures that will kind of describe it. So coming back, this is the books that I initially just kind of showed you. So this is trisomy 21, easy to detect on a metaphase or standard karyogram. You don't need any fancy testing for it. So you have extra copy of 21. Even if the patient has large gene deletions, for example, loss of chromosome at 5p or short arm of 5p, it is called Cray-Duchenne syndrome, you can detect it on a metaphase karyogram because it is a large deletion, so you can't really miss it. It's visible under the microscope. This is an example of a locus-specific probe, which is called the FISH testing. So you created probes, and each allele or each gene has two copies, so there should be always two signals for each. And if one is missing, you say it's deletion, or if there's one extra, you say it's duplication. So this is FISH. And this is what chromosomal microarray really pictorially looks like. So you have 23 pairs, 1, 2, 3, 4, all to an X and Y. And you have these specific, I really say it's hundreds and hundreds of FISH testing in one testing. So you have these probes against important regions on the chromosomes where we know if there's a deletion or duplication, a patient will have a phenotype. And then you compare the patient's DNA with the standard or a reference microarray. So again, this is a more detailed explanation of what a chromosomal microarray looks like. You have the patient's DNA, you have the control DNA, you put them both against the microarray chip that you have created at standard. We've already created this standard chip that has important probes against important regions of all 23 pairs of chromosome. And then you look for signal loss, as I said, deletion, or signal gain. Very simplistically, this is what chromosomal microarray is. And it will give you, say, for example, this is chromosome 7 plot, and the patient is this purple, and it is below the reference line. So it means there's a deletion on chromosome 7. So this is another way of looking at the microarray. Again, the signal, the report that we get, or when we are in the lab, that's how we get it. You get these signals and you say if the patient is above the reference or below. So it's duplication versus deletion. So the same testing, so this patient has trisomy 21. There's 3 ways of getting a diagnosis of trisomy 21. Hopefully, you can all diagnose Down syndrome clinically, but in order to confirm it, this is a metaphase spread. So this is the patient's metaphase. You have 1 extra 21. Say if you had ordered a FISH testing, you get 3 signals for the probe on chromosome 21, so that 1 extra. And then if you had ordered a chromosomal microarray at chromosome 21 probe, everything would be at higher noise or higher signal than the reference. All the other chromosomes in reference are at 1. However, chromosome 21 is higher. So even a chromosomal microarray can detect a trisomy, though it would not be a traditional way to order it, but you can certainly detect it if you were to order a CMA. So chromosomal microarray, really we use the acronym as CMA. So this cartoon shows here chromosomal aneuploidy. So aneuploidy means when you have extra chromosomes or fewer chromosomes, so like trisomy or monosomy, they both will come under aneuploidy. So you can detect an aneuploidy with both a karyotype and a CMA or a chromosomal microarray. You can detect a large deletion or duplication as well on a chromosomal microarray and a karyotype. The only thing that really a chromosomal microarray cannot detect that a karyotype can detect, which is important sometimes in clinic, certain very specific clinical situation, is if you have a balanced translocation. So if you remember, balanced translocation is a chunk of a chromosome from one kind of breaks off and sticks into the other chromosome, but the net genetic information in the patient remains the same, so it's balanced. So balanced translocation cannot be detected by a chromosomal microarray, because if you remember, chromosomal microarray only tells you signal from the reference, but because the genetic information is balanced, it cannot tell you if it's, it cannot tell you because there's nothing missing or duplicated. So I think that's an important point. Sometimes we get tested on this. So coming to sequencing. So sequencing has evolved a lot. So sequencing used to be done, well, it's still done in some situations, but you may hear about traditional Sanger sequencing. And then now we have essentially very large multi-gene panels, and we use what we call as next generation sequencing technology to build these multi-gene panels. And then whole exome and whole genome sequencing are again 2 other ways where next generation technology has been used to analyze exome and genome. So what is next generation sequencing technology? So I need to explain to you before we can kind of understand how that has been used for both exome, genome, and large gene panels. So you take a patient's DNA, we call it genomic DNA. You break that DNA into hundreds and hundreds of very small pieces. So when I say small pieces, they're about 100 base pairs or 200 base pairs, so very small. And then we already, as you recall, we already sequenced the human genome in 2003. So we know what the human genome is supposed to look like. So that is the normal reference genome, or not normal, I should say, it's a reference genome. And these small pieces of the patient's DNA that you have sequenced or broken down, you kind of align them against this reference genome, and you come up with a sequence. So that's the very simplistic way of how next generation sequencing works. You take a patient's DNA, you chop it off into hundreds and hundreds of pieces, you align it with a reference genome, and then you sequence and see what the patient's DNA reads compared to the reference DNA. And that's how you find mutations or variants. Now, how is exome different from genome? I think it's important to know. So exome is a very, very, very small piece, part of the whole genome. Exome is under 2% of a patient's genome. But over 85% of Mendelian disorders arise from that 2% or exome. So that's why our first step is usually whole exome if we have to do it, and we don't really jump straight to whole genome, though sometimes we do. So genome is obviously way more information or base pairs than an exome. but a lot of it you may kind of remember, we used to use the word junk DNA. So that's really what we meant by junk DNA. Only 2% of the entire genome is useful in its exome, though it's not entirely true. There is a lot of information even in the so-called junk DNA, but that's what used to be called junk DNA, and that's why exome is so small. This is another cartoon, is kind of my favorite. So if you think about the whole genome as an apple, then exome is really a small slice of that apple. And when we do whole exome, that's what we're looking at. So DNA is made up of exons and introns. Exons code for protein, so that's why they're important. We also know that 85% of Mendelian or genetic disorders arise from problems in the exon. Each exon is interspersed with an intron. So introns kind of are after each exon, but we don't know a lot of what introns do. We are starting to learn more and more, but that's how it is. There's more junk or non-informational DNA still, and we know less. Now, we talked about next generation sequencing technology. So next generation sequencing technology has been used to either do whole genome sequencing, or it has been used to do whole exome sequencing or target panel sequencing. So essentially why we use next generation and not Sanger is because Sanger is extremely slow, labor intensive, and probably way more expensive because it is so slow and requires really manual labor. So we really do next generation sequencing, labs, I should say, do next generation sequencing now. And when I say, what we mean by capture or no capture is if you really look at everything, so these colored rectangles are different exons and everything black is introns. So if you look at everything and you don't really pick and choose, you get your entire genome. If you only pick these colored boxes, these are the exons, you get whole exome sequencing. And then neurologists have become really smart, I should say, and we've created different panel testing. So we can do epilepsy gene panels or dystonia gene panels or ataxia gene panels. They're really commercially available. I know obviously they're available here, but I also know that even in India, many genetic labs have come up and they're offering testing. So, and that's called targeted gene panel. So you have curated a list of genes, and you're using the next generation to sequence all of them together because it's quicker. It's not that expensive actually to sequence many hundred genes together than one gene after the other. And I think in my second or third talk, I'll talk more about cost and those things. So genetic testing bias. So way of thinking is, should you look at the hay by a handful for anything that might be sharp, or should you look through the whole hay stack for the needle? And actually looking through the whole haystack for the needle is more cost effective, time effective, and is, I think, better in our field. So these are just some examples of how we have created so many different panels: a neuropathy panel, dystonia, Parkinson's, motor neuron disease, dementias, leukodystrophies, epilepsy, attacks, you name it, really. As long as you're comfortable with defining the phenotype, there is a gene panel available for it. So as long as the phenotype has been described correctly, I think it's easy enough to choose a panel. There is certainly a role for single gene testing with some very specific disorders, but more or less the phenotypes are tight, but the genotypes are so broad that we either do disease-specific next-generation sequencing, or in some situations even do whole exome sequencing as the first step. More so in pediatric neurology, I would have to say, though. Now, in terms of yield of genetic testing, why is genetic testing done? What is the yield or positivity in the right patient? So when we only had the carrier type available, the yield of genetic testing was really it was about 5 to 7%. So if you tested 100 people, you'll maybe find chromosomal disorders in 5 to 7%. That's it. That was the end of the story. With whole exome sequencing, it's still very modest. It's about 30 to 40%. So even now with everything that is available in the field of neurogenetics, the diagnostic yield can only be provided in the right patient in about 30 to 40% of the cases. The vast majority still, unfortunately, are not provided with an answer, but it is extremely high compared to the traditional testing of chromosomal analysis that we had really 15 years ago. So from 5% to 40%, I think that's pretty impressive. So in order to help you understand which has highest resolution, so karyotype certainly, as you know, probably makes more sense now, that has the lowest resolution. And as you go to FISH, CMA, which is chromosomal microarray, and then whole genome or whole exome sequencing, you increase the resolution. And you don't need any probes for whole genome sequencing. You certainly need a probe for FISH and chromosomal microarray. So that was another question that I had. So that was the first part of my talk. The other important point I wanted to highlight is, why do we do panel testing these days? There are two important concepts that are important to note. One is of what we call genetic heterogeneity, and one is what we call phenotypic pleiotropy. So let's understand what genetic heterogeneity means. So classic genotype-phenotype concordance means you have one genotype and you have one phenotype. So for example, patients with trisomy 21 or Down syndrome across ethnicities look exactly the same. You really, as a physician, should be able to see a child with Down syndrome and say the child has trisomy 21. There is no other chromosome that typically would mimic Down syndrome. So that's a very tight genotype-phenotype. It doesn't really happen that way in most genetic disorders, other than maybe some chromosomal disorders. So for example, you have a phenotype, you could have many genes that produce the same phenotype. A very classic example is something called hemiplegic migraine. So in hemiplegic migraine, patients' phenotype is pretty similar. They get hemiplegia and they get migraines, but their genotype could be very different. They could have a calcium channel mutation, they could have a sodium channel mutation, they could have an ATP channel mutation, or they could have PRTT2 mutation. Now, if a physician sitting in the office can diagnose hemiplegic migraine, but there's no way a physician looking at that patient with hemiplegic migraine can say which of these genes is mutated. So you have to do a panel. So this is what we call the concept of genetic heterogeneity, meaning the phenotype is caused by many different genes. Just the opposite is a phenotypic pleiotropy. So for example, you have one genotype, one gene, but it produces many different phenotypes. For example, a patient with a mutation in ATP183 gene, it is a channelopathy, it is a channel gene. It can produce 3 different phenotypes, as we know of today. It can cause alternative hemiplegia of childhood. It can cause rapid onset dystonia, Parkinsonism, and it can also cause infantile epilepsy. Now, you could have a family with this gene mutation, and different patients could have different phenotypes. There was no way you could have linked them to this gene unless you did either whole exome or a panel testing. So these are the two big reasons why in neurology and many other subspecialties, you really order panel testing and not one gene after the other, unless the phenotype and genotype was very clear. For example, a patient with neurofibromatosis type 1, there's nothing else that would look like that typically, and so you would just order NF1 gene testing. The other things to remember in exome sequencing, and this is more to do with the technology and how it's done, you cannot diagnose trinucleotide repeat disorders on exome sequencing. So trinucleotide disorders like Huntington disease or SCAs, spinocerebellar ataxias, you can't diagnose them. And for practical purposes, even large deletions or duplications cannot be detected on exome sequencing. So based on what we have learned in terms of technology and testing, this is how you would classify genetic disorders. So you have chromosomes, which are made up of DNA. You have genes on the chromosomes, and then genes could be, as I explained to you, the methylation of those histones can silence them, and they may not produce the protein, or they may produce the protein. So If a large chunk of chromosome is missing or there's some abnormality, this is called chromosomal disorders. For example, your trisomy 21. If a small piece of chromosome is missing or duplicated, for example, 22q11.2 syndrome, you call them microdeletions. So these are the disorders you can detect by chromosomal microarray. Now, if a gene doesn't function well, a single gene, you call it a single gene disorder. For example, neurofibromatosis type 1. And if the gene was supposed to make the protein, but for whatever reason it doesn't, we call them epigenetic disorders. So like imprinting disorders, Prader-Willi syndrome or Angelman syndrome. So those are classic examples of epigenetic disorders. So these are like 4 categories you could place most genetic disorders in. And then we also talked about the technology that can help you diagnose these disorders. I'll briefly talk about imprinting disorders because I think in neurology there are a few that kind of show up in boards. Prader-Willi and Angelman are classic examples. So DNA methylation is what really goes abnormal in these imprinting disorders. So DNA methylation, you have these cytosine residues in DNA that gets methylated, and when something gets methylated, it is silenced. So it doesn't make protein. So one classic example of this is X chromosome inactivation. You may recall that in females there are 2 X chromosomes and one of them is always silenced, only one expresses. And how that one X is silenced is by DNA methylation. And the other DNA methylation disorder is the Angelman and the Prader-Willi syndrome. And I'll explain to you with the help of this diagram because it can be a little tricky. So normally on the chromosome 15, there is a region. So this is normal. You kind of walk with me. So this is normal. This is a Prader-Willi region on the chromosome 15, and this is the Angelman syndrome region. Angelman syndrome region on chromosome 15. In normal cases, only one of these copies is active. The other is methylated. So on the maternal copy, the AS region is active, and on the paternal copy, the Prader-Willi is active. Now, when a mutation happens, that the paternal copy is deleted, that's how you get the Prader-Willi syndrome. And when a mutation happens, that the Angelman copy is deleted on the maternal, you get the Angelman syndrome. So these disorders kind of are extremely different. Prader-Willi has a very different phenotype than the Angelman syndrome, yet they're both caused by the same chromosomal disorder. And the reason is, in one case, the paternal copy is missing, in the other case, maternal copy is missing. So that's why it's important to know that you're both copies, you get one from dad and one from mom, and they're kind of important differently because many of the genes are only expressed from one and not from the other. Imprinting disorders are kind of tricky to remember. So I think as long as you understand that there's something called imprinting, and sometimes these disorders happen because of deletion on paternal copy or maternal copy because they function differently, I think that's kind of enough. Now we'll come to some basic inheritance modes. This is easy, hopefully. So we will focus on Mendelian inheritance. As you may all recall, Greg Mendel did some P experiments way back in 1856 from 1863, and he established the basic inheritance concepts or Mendelian inheritance. There are four inheritance patterns that we will talk about: dominant, recessive, X-linked, and mitochondrial. In the genetics clinic, patients, when they're seen, typically they should get a pedigree drawn out. If you may see some pedigrees and you will see that squares are males, circles are females. Anything that's colored means it's affected. Anything that has a dot means they're carriers. So not wholly affected, but they're carriers. And then there are other symbols that some of them you may want to know. If there are two lines between mom and dad, it means that they're related, so consanguinity. And then if there's an arrow for a box, it means it's the proband. So important concepts in autosomal dominant inheritance: affected individuals happen in successive generations because it's a dominant disorder. Both males and females are affected equally. New mutations may occur in an individual without affected parents. And so, even if there is no family history, you can have a new mutation. And there is variable expressivity within family. So, for example, I had that question on NF1 where the patient had all the features of cafe au lait and freckling, but mom only had maybe a couple cafe au lait and maybe 1 year of fibroma. So that is not incomplete penetrance, that is variable expressivity. So incomplete penetrance would be when you would not have any phenotypic feature, while variable expressivity means you have expression, but it is variable. And neurofibromatosis type 1 is a disorder that has complete penetrance, but it has variable expressivity. So that was something I discussed in that disorder, in that question. So the other group of disorders that as neurologists we come across is trinucleotide repeat disorder. So examples of them are Fragile X, Huntington disease, spinocerebellar ataxia, and myotonic dystrophy. And these disorders are all dominant disorders. And they do have something called anticipation, which means that each successive generation, the phenotype gets worse. So the disease starts earlier, and it could also be more severe. And the reason why the triplet expansion happens is because of error in DNA replication called slippage. So this is an example of Huntington's disease mutation. So because these mutations change with generations, we also sometimes use the term dynamic mutations, meaning that they change. So these repeats are unstable. Normally, a patient should have only equal to or less than 35 repeats of CAG in Huntington for Huntington gene. But if the patient has greater than 40, that's when the patient will express the disorder. And greater the number of repeats, earlier the age of onset. So as you see, the graph goes like that. So the number of repeats increases, the age of onset is less. So genetic anticipation, I briefly mentioned before, happens when trinucleotide repeat disorders due to instability result in increasing severity with successive generation. For example, this is a HD family, everybody who's colored is affected. So a lot of people affected, but as you can see here, even a child has symptoms. And in this generation, the onset was 22, alive at 64. So essentially it shows when it started and the patient died. Coming to recessive disorders. So recessive disorders, if you look at this pedigree, the half colors means they're carriers. So this patient is a carrier, this person is a carrier. They came together and they had a child who's affected, a child who's not affected, and then two children who are carriers. So greatest recurrence risk happens among siblings of affected individuals, certainly. Again, there's no gender preference. Males and females are equally likely to be affected. If parents are both carriers of the same recessive gene, then each pregnancy has a 25% chance of normal gene, 25% chance of affected, and 50% chance of being carriers. And again, consanguinity and ethnic background does influence the specific group of disorders because there's higher chances of two carriers coming together. So that was another question I had in the pre-test. Now coming to X-linked recessive disorders, this is another interesting group of disorders where both males and females can be affected, but females are usually very mildly affected just because of the phenomena of X-linked inactivation that I mentioned because of methylation. So females have two X chromosomes, but only one X is expressed, the other X isn't. So X-linked recessive disorders in females, they are called carriers, though they can have mild features of the disease. So incidence is much higher in males than females. All daughters, so here of an affected male will be carrier, because as you know, the female will get the X chromosome from the dad. So the affected dad will pass the affected X chromosome to the female. And the condition, again, when you look at the pedigree, and if the condition can never be transmitted from father to son, so that's another clue for X-linked transmission, because the son would get the Y from the father and the normal X from the mom. So the son would never have an X-linked disorder transmitted from father. Now, a few examples pertinent to neurology I kind of listed here that you may know are X-linked is Fragile X, Duchenne's muscular dystrophy, X-linked adrenoleukodystrophy, or AMN adrenomyeloneuropathy, which is again the adult version, and even females present with AMN. Rett syndrome is X-linked, and then Hunter's disease, which is a lysosomal storage disease, is X-linked. Actually, one very important disease that's missing, and this is Fabry's disease, which is also X-linked and can present with strokes in young people. So coming to the last group of Mendelian disorders is mitochondrial disorders. So first, before I go into the inheritance of mitochondrial disorders, I thought I'll spend a few minutes discussing the mitochondria. So mitochondria, as we know, are the principal source of energy for all cells except RBCs. Each cell contains hundreds of mitochondria, and the mitochondrial DNA is really very small. It really has only a small DNA that makes 37 genes. So mitochondrial DNA in itself is very small, but there are over 1,000 gene products in the mitochondria. So rest of them are encoded by the nuclear DNA. I think that's an important concept to remember. And I have this slide that tells you how many genes are involved in mitochondrial machinery. So you have this mitochondrial matrix, you have outer mitochondrial membrane, you have inner mitochondrial membrane, you have the intermitochondrial space, and then so many other proteins. This is just a very modest list of proteins or genes that make proteins for the mitochondrial machinery. But as I showed in the previous slide, only 37 of greater than 1,000 are encoded by the mitochondrial DNA. So why this is important? So mitochondrial DNA is what is transmitted in a mitochondrial inheritance fashion, but the nuclear DNA is Mendelian, so it's mostly recessive. So most mitochondrial disorders are actually recessive inheritance and not mitochondrial. So that's where this concept of dual genome origin comes in. So dual genome means the mitochondrial DNA machinery or mitochondrial machinery is run by this nuclearly encoded DNA and mitochondrial DNA. So this follows, the mitochondrial follows the maternal inheritance that we classically know of as mitochondrial DNA, but the nuclear will follow Mendelian. So it could be recessive, dominant, X-linked, but most of the time it is recessive. Mitochondrial DNA is exclusively inherited from the egg. There is no contribution from the sperm, and hence this is how a mitochondrial pedigree would look like, that if a boy or a male has a mitochondrial disease, they cannot pass it on to their children, while if a female has a mitochondrial disease, because they pass on abnormal mitochondria to all their children will be affected, though again the severity could be mild to severe. And here, the severity depends on something called heteroplasmy. So heteroplasmy is, again, an important concept for mitochondrial inheritance. So let's kind of walk through this graph here. So this is a mom. She has very mild symptoms, or let's say no symptoms at all. The red mitochondria are mutated mitochondria. So mitochondrial cells may have 20% mother cells may have about 20% of mutant mitochondria, and yet she may not have symptoms. So this is mom's DNA. As eggs are being made, these red or the mutated mitochondria may pass on in different numbers. And if you know, mitochondria are autonomous, so self-replicating. So a cell with fewer bad mitochondria will end up having less amount of mutant mitochondria, while a cell with greater number of mutant mitochondria will have more. So this is called heteroplasmy. And so a child could inherit 80% of mutant mitochondria and they would have severe disease, while a child could have only 20% of mitochondrial disease, mutant mitochondria and would have no disease. So this is called heteroplasmy, which is essentially a way we tell you what is the ratio of mutant mitochondrial DNA to normal DNA. And it actually can vary from cell to cell. We can check it on the tissue that's sent for testing. So for example, a mitochondrial patient has blood testing. You could tell them what is the heteroplasmy for WBCs, but that doesn't mean that their cardiac cells or the brain cells or liver, whatnot, have the same amount of heteroplasmy. It's really tissue specific. And so that's how sometimes the patients with very low heteroplasmy in their blood still could have severe symptoms in their liver or brain. But this is a rough estimate of how many mutant mitochondria they have. So that's the end of the mitochondrial inheritance. Now there's also something called multifactorial inheritance. So most disorders that we deal with as physicians in the clinic are multifactorial. Mendelian are few. So multifactorial, there's an interaction of genetic factors, environmental influences. And so we think of medical disorders being as a continuum. So there are some disorders that are purely genetic. For example, we talked about Huntington disease or Fragile X and things like that. And then there are disorders with some environmental influence. For example, patients could have predisposing genes for dementias, but say they change their environment or disease modifying things and they may not develop it. However, then there are disorders that are purely environmental. So I think this is a spectrum, but these are some examples of multifactorial inheritance. So they could be mental illness, diabetes, MS, dementia. They're all kind of multifactorial for most part. I will skip that. The other, I had this slide on pre-implantation genetic diagnosis. So I think one question comes is what can be done sometimes with this genetic information. So the newer technology that is used really quite commonly now for many severe genetic disorders is something called pre-implantation genetic diagnosis. For example, if there is a family, say with, let's say Huntington disease, that it's a dominant condition, so anybody who has it has a 50% chance of passing it on, but they want to choose not to pass it on and they want to have children the normal way. So essentially you offer them what we call as pre-implantation genetic diagnosis, so something similar to in vitro fertilization. So they go through that process of in vitro fertilization. And then once the embryo is at the blastocyst stage, you remove or you check for the DNA mutation and then only implant the embryo that doesn't have the DNA mutation. So that's one kind of application of genetics early on where you may be able to avoid passing on a severe genetic disorder in the family. So I think that's kind of what I had for today. Genetic diagnosis and inheritance. I'll stop here. If we have time, we can, I think we're kind of over time already, so we can not do the after questions or the pre-test questions again. I'll take any questions if anybody has or.
Sudhir, hi, I'm Roop Gursahani. I'm waiting for Sudhir to appear.
Oh, okay.
Can you hear me? Yeah. Hi. So there are some questions on the chat. I'll quickly run through them. Do you want to open them yourself? Or one is NGS's second generation sequencing with Sanger being the first. What is third gen and is it still commercial? Is it already commercial?
Okay, let's see.
Did you see it?
Yeah, we typically use second generation or next gen sequencing clinically. I'm sure there are more sequencing technology that are in research, but not commercially available. I think that kind of answers that question.
Is Sanger really, I mean, is there any advantage to doing Sanger? I mean, if you look at all the manual effort, is it really Is it?
I think Sanger sequencing has a place. So sometimes when we do next generation sequencing and we find variants, sometimes we use Sanger to actually confirm that those variants are real. And Sanger sequencing is also used, for example, you're only ordering a single gene testing, for example, as I give an example of NF1 or neurofibromatosis 1 testing. So when you order testing, they're not going to do next gen for that. We can order Sanger sequencing for single gene. So I think it still has a place. But for vast majority, I think we're all into next generation sequencing. Which is more confirmatory, Sanger or next generation? You know, I mean, as I said, both have their time, both have their place. To confirm next generation, we sometimes use Sanger. Sanger is, it is confirmatory. If you see a variant in Sanger, it is a true variant. But in next generation sequencing, because of some limitations, you may have to do Sanger to confirm it. So out of the two, I would say both are confirmatory, though Sanger is gold standard.
Okay.
Yeah, I had one question. There is the concept that the mitochondria are actually bacteria which have gone into the cell. So they are different sort of organism which is living inside each cell. So how did the bacterial DNA get into our nuclear DNA? I mean, so why is it not completely separate?
Yeah, I'm not sure if I have the answer to that question. I will have to do some reading around that.
Okay.
That happened a long time ago.
Yes, long time ago. But still, I mean, now they are blaming the mitochondrial DNA for excessive immune response. So when you have some, if there is some tissue damage, then the mitochondrial DNA gets exposed, and then that causes a lot of inflammation. So they are blaming it in all sorts of these inflammatory responses also. So the mitochondrial DNA is becoming very important.
Okay, so I'll just quickly run through the questions. I think there's only one more which I thought really needed to be answered because that's really very specific. Notating hyperlipidemia in pedigree. What is the role of inserting a small square in the corners of a pedigree square or circle? Yeah, I think I've also seen that once. Can you?
Yeah, so if there are many disorders, so sometimes we put like a small square that you could actually make your own key. So you could make a small square, say it says hypertension or hyperlipidemia, and the other circle in the other corner. So each corner could be one disorder. Does that make sense? So if it's a left corner, it means hyperlipidemia. If it's the right corner, it could mean diabetes or things like that. So when there's so many, I don't think there's a rule for which corner it can be. You can make your own key as long as the key and the pedigree match. It works.
So basically, I mean, this is the language of the genetic clinic.
Yeah. It's
all
the
of
language
us
of hopefully they will simplify it in their notes that you can read it. I don't think neurologists are expected to read pedigrees.
Okay, a little difference between epigenetics and single genetics. I mean,
Mendelian. Yeah, so epigenetics exactly is an example where when gene is made into proteins, so the example of epigenetic disorders is methylation defect disorders like Prader-Willi and Angelman syndrome, where there is a methylation defect in how the gene is expressed. So it's not the mutation in the gene, but how the gene was processed. So a step after that. So single gene disorders say there's a sequencing error. So you're reading the NF gene ACTG and T is missing or C is missing, that is a single gene disorder. But if the histone code is a separate code, if there's more methylation, less methylation, so when there's a methylation defect, those disorders are considered epigenetic disorders. And the classic example would be, in my mind, Prader-Willi and Angelman syndrome for our purposes.
Not so big in neurology as compared to oncology, for instance, I guess.
Correct.
What controls the epigenetics thing? I mean, so the gene expression is controlled even later after the gene is formed by the epigenetics. So what controls that?
Yeah, there are many factors. So I think one big thing we can all know that controls is something called microRNA. They're very new fancy term in DNA language. So if you want to read about microRNAs, you can learn a lot about how gene is regulated and controlled. So I would say that as a good example.
I mean, isn't that epigenetics is to do with turning off and on genes at various times and
you got it.
regions?
You got it. Yeah. So different genes can express less or more at different times in a life cycle, or in different tissue specific. So exactly that. Yeah. But I think those are more, as you maybe said, maybe in more cancer or acquired. While when we're talking about inherited or Mendelian disorders, then there are very few examples for those.
Okay. Now I appreciate that it's really late for you on a Saturday evening. The last, I think I'm going to ask this. Would you want to explain linkage analysis in short and kind of we stop at that?
Yeah, so linkage analysis is a technology or technique that is used for gene discoveries. So the simplistic way of thinking about it, you're trying to find an unidentified or a new gene, but you know the genes that are before or after, so they're kind of linked together. So that's really what linkage analysis is. It's used mostly for gene discoveries because you're suspicious of a specific locus, you make probes for around, so they're kind of linked together and you try to study them in inheritance. So that's where its application is. In our clinical field, I think we're way beyond it. We can sequence the genome now, so we really don't need linkage analysis. But for gene discoveries, that's what we use, or it was used.
I have not understood incomplete penetrance versus incomplete expression. Which diseases have incomplete penetrance?
So a lot of adult onset neurodegenerative disorder genes, for example, dementia genes, so like presenile 1, 2, frontotemporal dementia genes, those are classic examples where you could have a gene mutation, but you may not have the disease. So they're incompletely penetrant. But neurofibromatosis type 1, tuberous sclerosis, all these neurocutaneous disorder genes are completely penetrant, though their penetrance is age dependent. So a child may not have all the manifestations of NF. They may only have cafe au lait or freckling, but as they get older, they get more neurofibromas. So it's age dependent penetrance, but they're completely penetrant. But that's variable expressivity where you have different features, or mild versus severe.
Okay.
Done.
Okay, thank you.
That was lovely. I mean,
that was very nice.
So, okay, I'm glad you enjoyed it. Okay, yes. Thank you. I'll see you next Sunday.
I think to explain something nicely requires knowing it yourself very well, and you really told us how well you understand it. When you explain it so simply, it makes us think that we also know genetics.